for accepting wholeheartedly to conduct this 47th International Virtual Learning Series with the blessing of Almighty, our founder Kalvi Valan Sri Kalasalingam Ayya, and with the support of our chairman Ilayvalal Dr. S. Sridharan sir for the 31st International Virtual Series titled on Engineered Nanomaterials for Real Life Application and a Research Life in association with the Indian Pharmaceutical Association South Tamil Nadu Local Branch, Head Office at Arulmigu Kalasalingam College of Pharmacy. Today we are proud to have an eminent scientist, notorious researcher, distinguished personality, and our honorable resource scientist, Dr. R. Mohankandhasamy, Waterloo Institute of Nanotechnology, University of Waterloo, Canada, USA. To grace this occasion, it is a great honor to have with us for this occasion. We thank you for kindly being a valuable part of this session, sir. Let me appear to you with sterling quality of this eminent personality, a brilliant researcher. Sir, your prestigious presence is a great honor for us and great motivation and inspiration for our entire student community. Sir, we are fully appreciate you for finding the time to be with us despite of your busy schedule, sir. Most welcome, sir. Andasamy, sir. Most welcome, sir.
Thank you, sir.
Thank you, sir. Thank you, sir. I want to extend my sincere thanks to management of AKCP for accepting wholeheartedly to conduct this international virtual learning series. It is gratifying to note that this session covers a wide range of interesting topics relating to the engineering nanomaterial for real life application and a research life in abroad. So I hope this session will be a good platform for our students. Dear students, kindly utilize, grab this opportunity. This is the first and foremost needy for your career. Finally, I welcome all the professors, faculty members, and my dear enthusiastic students for today's session. All the very best, my dear students. Once again, I welcome you all. Thank you. Most welcome, Dr. Mohankandhasamy, sir. Most welcome, sir. Thank you, sir. You can proceed, sir.
Thank you, sir. First of all, I want to extend my thanks to Professor N. Venkatesan, Principal Sir, AKCP, and the management of AKCP to providing me an opportunity to deliver a webinar lecture today at this time. Thanks for the faculty members, especially Dr. Anurag sir and other faculty members joining with me today, and most importantly, my dear students. Thanks for joining me here. So let's get started. So today we are going to see about the topic on engineering nanomaterials in real life applications and research life in abroad. So in this lecture, I'm going to talk about how the nanomaterials are fabricated to use for real life applications, and important topic is how to manage or how to live a research life in abroad. Myself, Mohankandhasamy, completed B.Pharm, M.Pharm from KMCH College of Pharmacy, Coimbatore, and I'm working as a postdoctoral fellow in University of Waterloo right now. Today's agenda, we have three parts. First part, we are going to see about what are the surface engineering of metal nanoparticles for biomedical applications. In that, I'm going to explain about what is photothermal therapy and what is antibiofilm therapy using surface modified nanoparticles. In part 2, I'm going to give a brief about what are sustainable nanomaterials and how they could be applied in dental, cosmetic, and probiotic real life applications. And as the last part, I'm going to explain about what is research life in east and western part of this continent, means Republic of Korea and in Canada. So moving on to the presentation, the part one, I'm going to cover the biomedical applications. So surface functionalization of nanoparticles. So you guys might have heard about nanoparticles, you might have read about nanoparticles, or you might have been doing research on nanoparticles right now. So what are nanoparticles? You are well known. So nanoparticles are different according to its size, surface, shape, and material. So the nanoparticle size ranges from 1 nanometer to 100 nanometer. Depends upon the size, their application and activities are entirely different. And according to its surface, again, their applications are entirely different. As shown in this picture, we can modify its surface by different chemical and biological molecules, and especially the shape of these nanoparticles. Yes, definitely, they also play a major role in the application part. I will explain in detail in the later of this presentation. And the another important factor is the material. What type of material the nanoparticles are made up of. So it varies from inorganic nanoparticle, polymeric nanoparticle, and so on. The composites like combination of inorganic and polymeric nanoparticles and uh, plant-derived nanomaterials like chitosan nanoparticles, etc. As shown in this picture on the right-hand side, the nanoparticles can be surface modified into these different categories, are like molecules. So see, here is an example. This is a hydroxyl group containing nanoparticle, and it can be modified using different techniques to contain silica group or aldehyde group or acid group and etc. Further, they can be modified with the other chemical or biological or other molecules as shown in here, or even nanoparticles as shown in here. So it may be a protein or antibody, it may be alkyl or aryl group, it may be DNA or siRNA, it may be a biotin, or it may be another nanoparticle, and etc. So again, see, the surface functionalization of nanoparticle is a wide range of topic. It has wide applications. So it is an important tool to understand, to know what you're going to develop as your nanocomposite. That's the important part. So before going to the application part, you have to familiarize, or you have to be very well versed about what you're going to modify the surface of the nanoparticles. So according to the modified surface, the nanocomposites can be applied in the following areas, starting from textiles, biomedical, food and agriculture, industrial, electronics, environment, and energy applications. So as shown in this figure, the nanoparticle applications are day by day, their horizon is expanding largely. For example, in the case of biomedical, these modified nanoparticles could be used in cancer therapy, drug delivery, and in UV protection as sunscreen. In food and agriculture, the nanoparticles could be used as a food packaging materials. In the case of industrial purposes, they used as a photocatalyst or industrial catalyst to uh, remove the waste materials like organic dyes, especially. In the case of electronics, yes, due to the development of nanoparticles last 2 decades, the all electronic devices becoming very smaller thanks to the nanotechnology. This plays a major role in this part of electronics. And environment, yes, nanoparticles also play a major role in environmental impacts. Apart from environmental impacts, they could be used as in environmental applications like wastewater treatment to separate waste in the water, make it as a drinkable water, and catalysis. In energy, as you all know, the solar cells are made up of nanoparticles, and they are providing enormous energy source throughout this globe. Lastly, textiles, yes, there are a lot of surface modifications and a lot of developments have been happened this year. So that's why the nanoparticles are being applied in textiles like intelligent textiles, or you can call as a modern or sensor-based techniques textiles because of the nanoparticles. So these are the different applications of surface-modified nanoparticles. And I'm going to talk about gold nanoparticles, especially why we chose especially gold nanoparticles when I was doing my PhD back in Republic of Korea, I mean South Korea. We chose to work on gold nanorods. Why gold? Gold is an inert biocompatible material. It has peculiar, specific, unique characteristics called surface plasmon resonance. None of the other nanoparticles except silver has this property. Because of that, they can absorb the light and convert that into a heat. That's the very peculiar advantage of silver, gold nanoparticles. So, gold nanoparticles, according to its shape and size, they they behave differently. So here you can see spherical gold nanoparticles, which are all 10 nanometer in size, elongated form of gold nanoparticles called gold nanorods, gold nanostars, gold nanocubes, gold nanocells, and gold nanoclusters. So why there are different varieties or different shape of gold nanoparticles, although they are made up of same metal called gold. According to the purpose of its applications, we can modify or we can synthesize the nanoparticles to its different structures. So you will see why we worked on gold nanorods in the upcoming slides. So section 1 about my first PhD thesis title, we prepared magnetic optical gold nanorods for recyclable photothermal ablation of bacteria. In simple terms, we prepared gold nanorod containing magnetic nanoparticle that could be used as a recyclable antimicrobial agent using laser light. As shown in the right hand side picture, the gold nanorods structure initially like this, and they could be modified with the different chemical moieties. Later, it could be attached with the magnetic nanoparticle to make it as a magnetic gold nanorod. So then those gold nanoparticles were treated with the pathogenic bacteria and passing the laser light near infrared laser at specific wavelength. They got heated and they kill the pathogenic bacteria as shown in this picture. And those nanoparticles could be reused like 5 to 6 times with the same temperature, with same antimicrobial property. So this idea was created like AquaGuard in India. So we used to change the cartridge, right? The similar way we were thinking to keep this nanoparticle inside the cartridge and passing the laser light. Those cartridges which is having nanoparticle get heated because of the laser light, and it can kill the bacteria. That was the concept. So these pictures shows the microscopic images, fluorescent microscopic images on the left hand side and the scanning electron microscopic images on the right hand side showing the destruction of the bacterial surfaces. So this work was done in the Gachon University, Republic of Korea. So moving forward, we did the characterization of gold nanorod and magnetic nanoparticles. As you see here, the gold magnetic nanoparticles, which are made up of iron oxide Fe2O3 nanoparticles, they are covered with silica to make it as a silica coating silica coating magnetic nanoparticles. Why we need to coat silica? Because iron oxide nanoparticles are easily oxidized because in the presence of oxygen. So to prevent the oxidation, we cover that with silica. Then this is the transmission electron microscopy images of gold nanoparticles, gold nanorods, sorry. So these techniques are characterization techniques which are used to find out the nanoparticle shape and its surface modifications, especially the EDX. It's called energy dispersive X-ray spectroscopy. It will provide the details of the material what you have on your sample. So if you click your mouse in this machine, it will show what is the composition of the material. So as you can see here, it has gold, silica, Fe, and copper. Copper is the grid used to prepare the sample. So we can confirm that our sample, the conjugated sample gold nanorod containing magnetic nanoparticle has gold silica and iron Fe. In this way, we could confirm the successful conjugation of the nanorod on nanoparticles with nanoparticles. Sorry. This is the UV visible spectroscopy of gold nanorod. Once the gold nanorod surface is modified, the lambda max of this gold nanorod's absorbance core redshifted. It means that the surface is modified with some chemical or physical or other nanoparticles. So that way we could confirm also. So those gold nanorod containing magnetic nanoparticles were treated with bacteria, pathogenic bacteria, gram-negative and gram-positive bacteria to find out the efficacy or photothermal efficacy of these nanorods. So when the laser light and a near infrared laser source, when the laser light's power was increased to 10 to 130, it produces better antimicrobial property. This is the fluorescent microscopic images. These green and red dots are bacteria. The green dots are alive bacteria. The red dots are dead bacteria. There are special stains. We can use those stains to differentiate live and dead bacteria. So those stains called live dead bacteria dye. So if the bacteria is alive, these dyes can attach with the DNA to produce green fluorescence. So it's in that way we could differentiate green and red cells. In that way we can say they are dead, the cells are dead. So at 130 milliwatt, the nanoparticles were completely eradicated. This means that our nanorods are highly antibacterial in nature in the photothermal therapy. So that's the first part, in the first section. In the second section, we did biofilm formation and antibiofilm assays. So what are biofilms? Biofilms, as you all know, bacteria are available everywhere and especially in the liquid, especially in the water, liquid media. Those are called planktonic bacteria. Once the bacteria attached on surfaces and when it started growing a mature structure, they are called biofilms. So this is the cycle of biofilm. Initially, the bacteria started attaching on a surface. Mostly uh, bacteria love hydrophobic surfaces, so they start attaching on the hydrophobic surfaces. Then they start growing. They start forming exopolysaccharides and some enzymes and etc. They started forming a biofilm growth. So once the biofilm are got mature, they are called a 3D structure of the biofilm, which is covered by exopolysaccharide membrane. So this exopolysaccharide membrane is a kind of a barrier to prevent the bacteria from the external surfaces like antibiotics. So why biofilms are important? Biofilms are 1,000 times more stronger than free-flowing bacteria, free-floating bacteria, or planktonic bacteria. So 80% of the hospital-acquired diseases are spread by biofilms. So biofilms are hard to treat against by antibiotics. So the biofilms are given much importance, especially biofilm inhibition. And those grown biofilms started maturate and they started spread, disseminate into the for the next cycle. So this is the life cycle of biofilms. So this and these biofilms are, as you can see in the dental plaque, wound biofilm on the surface of the wound and catheters and other biomedical devices and pipelines. These biofilms are looks visibly like this. When you look under the confocal microscopy, biofilm surfaces are looks like this. So this means these are the mature biofilms. They are very strong against antibiotics. Antibiotics cannot act against these biofilms. So our aim was to inhibit or reduce the biofilm formation. It means it will kill the biofilm cells. That's called persistent cells. Those are all much stronger against any antibiotics. So our aim was to reduce the biofilm inhibition by inhibiting the biofilm growth. Moving forward, the nanotechnology gave better solutions for biofilm inhibitions or biofilm treatment, I say. So this cartoon shows the left-hand side, what are the possible infection sites for biofilms in the human body, starting from eye, mouth, respiratory tract, endocardial infections, biliary tract, urinary tract, bone-related infections, and wound infections. On the other side, you can see what are the nanotechnology-based solutions we can provide. So, nanoparticles could be used to formulate contact lenses, oral implants, endotracheal tubes, heart valves and pacemakers, urinary catheters, especially prosthetic joints, and wound dressings using silver nanoparticles in bandage as an example. So these are the possible ways we could treat the biofilms for in the case of clinical applications. So how did these nanoparticles work on biofilms? So these are the different mechanisms we could formulate. So the nanoparticles could be used to make an anti-adhesive nanoparticle surface like super hydrophobic surface where the bacteria cannot stick and grow. It means the bacteria could be repelled if we try to attach on the surfaces. In the case of the other part, we can use polymeric crafted biocide nanoparticles. Means polymers can be modified to form as antibacterial polymers. Next, we can make bioactive nanoparticles. Means we can attach any drug molecules like plant extract or drug or highly antibacterial drug to make it as a bioactive nanoparticles. And next, we can make ROS producing a nitrous oxide releasing nanoparticles. There, those nanoparticles, when the bacteria tried to come in contact, those nanoparticles started producing gases to kill the bacteria. As last part, there are stimuli responsive nanoparticles could be made. These nanoparticles using ultrasonic waves and laser lights, they got activated and they kill the bacteria. That way purify the surfaces. So these are the different ways we can control the bacterial infection, obviously biofilm formation. So as the part of that, we are trying to prove our hypothesis. Yes, nanoparticles are the best antibiofilm agents. So we did this experiment. Cinnamaldehyde, as you all well know, this cinnamaldehyde is extracted from the cinnamon bark. So you can see a number of research articles available claiming that plant extract reduced silver nanoparticle, gold nanoparticle can be used as an antibacterial agent. But most of the papers cannot say exactly which molecule of the plant extract is responsible for the formation of nanoparticles. So we want to make it in a different way, like we used pure cinnamaldehyde and we check whether those cinnamaldehyde can able to form nanoparticles or not. Yes, cinnamaldehyde can form nanoparticles in a simpler way. Just mix a cold solution, cinnamaldehyde solution, just heat it for about 70 to 80 degrees in seconds, like 2 to 3 seconds, it will form cold nanoparticles. You can see the color change to pink color, means the formation of cold nanoparticles. And those cold nanoparticles were tested for its antibiofilm activity. So, characterization of the synthesized nanoparticle. As you know, this is the UV visible spectroscopy. So, cinnamaldehyde shows peculiar lambda max around 290 nanometer and 230 nanometer. When those cinnamaldehyde was used to form gold nanoparticles, first the solution becomes wine red in color. Next, it brings a unique peak around 530 nanometer. This unique peak is only a characteristic peak of gold nanoparticles. Usually silver nanoparticles will get around 440, I guess. Yes, this is gold nanoparticles around 550, 525 to 550 according to size. We can say the gold nanoparticles are formed, and most importantly, they are having containing cinnamaldehyde on its surface. So this gold nanoparticles were after purification, like after removing the excess cinnamaldehyde. So this is a kind of a confirmation. Further, as I said before, transmission electron microscopy. These are the electron microscopes used to find out what is in the surface of the nanoparticle. So this black round shaped one is metal nanoparticles. So metal under the laser beam of the electron microscope, they act as a contrast agents, like they absorb more electrons and so emit as a dark color. So in that way we can confirm this is the cold nanoparticle. Size was about 15 to 20 nanometer. And if you see this inside, you can see there is a layer around the gold nanoparticle that is cinnamaldehyde. Since cinnamaldehyde was used to form nanoparticles, later cinnamaldehyde act as a stabilizing agent to prevent the gold nanoparticles to degrade further or to prevent it from the aggregation. So moving forward, these gold nanoparticles are used to kill the pathogenic bacteria, especially gram-negative, gram-positive, and antibiotic-resistant bacteria. As you see from this graph, at very lowest concentration, the nanoparticles were highly active and they eradicate the biofilm, which means that the nanoparticles can penetrate through the surface of the biofilm and it can kill the bacteria. That's the mechanism we found out. As a part two, we are moving forward to see about what are the real life applications of sustainable nanomaterial. Sustainable nanomaterial means what are the nanoparticles are obtained from natural resources like from plant sources, for example. So in our lab in University of Waterloo, Department of Chemical Engineering, Institute of Nanotechnology, we are working on cellulose nanocrystals. What are cellulose nanocrystals? Cellulose nanocrystals are nothing but nanomaterials obtained from plant sources. So these are the different sources of plants. The stem of the plant was cut down and it was started hydrolyzed using sulfuric acid, strong acids, mostly sulfuric acid, and it was uh, divided into hemicellulose and lignin first, then further hydrolysis brings cellulose fibrils. Hydrolyzing further will provide microfibrils, and hydrolyzing further from there, it produces uh, cellulose nanocrystals by leaving amorphous region as a waste. So the crystalline region was extracted and is called cellulose nanocrystals. Why cellulose nanocrystals are important? Because of its properties. According to the acid we use, we can get the surface of cellulose nanocrystal having sulfonate groups, hydroxyl groups, carboxyl groups, and aldehyde groups. And it can be modified as cationic or anionic charge. So the physical properties of cellulose nanocrystals, they are high in surface area, they are crystalline in nature, they are more rigid, means more stiffer than steel, I say. And they can disperse well in the water, and they have unique characteristics called biorefringence, means when the light is passed in different angle, according to the concentration of the nanoparticle, they show different color. So where the cellulose nanocrystals are being applied. So cellulose nanocrystals are obtained from the plant sources in different forms, and they could be used as a nano or micro scale, as shown in this picture, like transparent film. And further, these days it is applied as a smart materials like transparent 3D printing machines, thin films, photovoltaic cells, biosensor for bacteria or for pathogens, even for viruses like COVID-19, and smart paper, and etc. So these are the applications of cellulose nanocrystals. So data about cellulose nanocrystals are called CNC. CNCs, every year the publications are increasing. Like as of 2017, according to the Web of Science report, up to 1,000 papers were published. Surprisingly, more than paper publication, the patents were filed every year. So it means that cellulose nanocrystals are being used in real life applications a lot. So using cellulose nanocrystals, we are developing anti-carry or antibacterial dental materials. So this is the background about that study. So oral diseases are non-communicable diseases, and most of the people in this world are affected. Like half of the world population is affected by the oral diseases, including dental caries. There are available materials as shown in here, starting from amalgam, cements, impression materials, and preventive materials are available to prevent dental caries and dental infections, I say, but they are all having disadvantages. Usually amalgam was used before days. It has highly antibacterial because of silver, copper, and mercury, but unfortunately mercury causes neurotoxicity and it shows poor refractive index. You may see olden days people has some black color teeth. It is because of this mercury. So this is not practiced these days, but still the other forms like polymers, resins, matrices are being used as a replaceable dental materials, but still they are also suffering from the following disadvantages. They are not stable for a longer time. They cause secondary caries. The polymers will shrink in time pass by, and it will form wear and tear on the teeth. So these are the disadvantages. So we want to provide a solution for these disadvantages. So we are developing kind of cellulose nanocrystal modified its polymer and its surface was made as an antimicrobial. So how this agent going to be work after filling on the teeth? These polymer chains will be extended outside. So when the bacteria come in contact with the quaternary ammonium group, the bacterial layer membrane is destabilized and the bacterial membrane is degraded, thus brings the death of bacteria. So this is our mechanism, and we are working on it to develop a better dental antimicrobial agents. We got best results due to the issues. I cannot show it here, but yeah, we have some comprehensive or better results. So what's the purpose of the material? So cellulose nanocrystals can be modified with polymer as a blue line, uh, plus as a quaternary ammonium group, pink dots as a nanoparticles, and green dots as a plant materials like cinnamaldehyde. So these could be used as an antimicrobial agent in photocatalysis to purify water, imaging agent because metal nanoparticle, and could be used in cosmetics. Further, according to its different surface modification, it can be used as a thermoresponsive drug delivery system because of the polymers. The polymers what we are working on, they are thermoresponsive. If you heat it, they become aggregated. If it is become to room temperature, they are got extended. The polymer chain will shrink when they got heated. When it cool down, the polymer change like extend back into its pendant or free floating. In photothermal therapy, because of the nanoparticles, it could be used as a photothermal therapy. Because of quaternary ammonium group, it can be applied as a gene delivery. And again, it could be used as a produce coating, like vegetables and other fruits can be coated with the cellulose nanocrystal material to prevent them from further degradation. So these are the different applications of the cellulose nanocrystals we could expect in the, or we are still working on it. Moving forward, we are working on a different project using cellulose nanocrystals, how to make a skincare product. So what are skincare products? As you guys are using every day or every other day with different skincare products, especially girls and women, there are ranges of skincare products available, starting from face cream, serum, hand cream, sheet mask, sun care. So all these skincare products, they are using a lot of chemicals, synthetic chemicals or modified chemicals, etc. So those chemicals are not good for our skin. According to the report, every American woman is using 200 chemicals on their face in one day. So just imagine how much chemicals you guys are using on your skin. So we want to bring a solution for this. So these further slides will give more explanation. So global comparison, who consumes more cosmetics in this world? Without a surprise, America is in number one and Japan shares number two. In third place comes China. Fourth place, surprisingly, it's India. Fifth place is South Korea, and sixth place is Canada. Why I said India is surprisingly? Because still we are we think that we are still using our traditional things for our beautification purposes, but uh, due to the global market and due to the globalization impact, India is a kind of largest consumer of the cosmetic products are the international brands. So that's why India is at fourth place. So Canada is in 16th place because Canadians believe more natural based ingredients. So because of that, there are not much cosmetic products available in that formulation. So that's why Canada lies under 16th. Canada consumes less cosmetics than India, than other countries. So this is the big or huge market. That's why we are focusing on this market. Moving on next, we are working on sunscreens. We are developing sunscreen creams without any chemicals. That's our ideal goal, our initial goal. So how the sunscreens work? Take an example in this diagram. It's a skin. The blue part is sunscreen. So sunscreen can act as a two different ways. research topic, so I'm going to my experience or general experiences. So I request all of you students, please listen. These steps might be useful because when we were doing BPharm, we were not this much blessed to have advanced technique or advanced knowledge or advanced experienced personals to guide us. Although our pharmacy teachers were best, but still we were limited, our knowledge were limited, so that's why we were struggling. But you guys have a great platform in these days, especially through these kind of webinars. So please utilize it, and try to move forward in your life. As a pharmacy student from India, especially from southern India, you are all well competitive, and you are all in professional degree. So you can compete in any part of this world. So keep that in mind. So please listen following things. So what is graduate study? Which are all the studies containing thesis based convocation? They are called graduate study, like M Pharmacy and PhD mostly. Although pharmacy in India we have undergraduate project, but they are not still considered as a graduate study in the case of Western culture. So what you need to do to apply for a graduate courses in foreign country, first you need to fix your specialization. So if you are doing your M Pharmacy or you are planning to do your graduate study in for other foreign country from your B Pharm, you have to start think about which specialization you are good at. So you can take that point from your curriculum marks, or you can your inner feeling will know you studied 4 year of pharmacy study, so you will know which subject you are good at it. So you can go with either pharmaceutics, pharmaceutical analysis, pharmaceutical chemistry, pharmacology, pharmacology, and etc. So first, fix your specialization, fix your goal, and that specialization should be your lifelong specialization. Especially in the foreign countries, once you started doing your graduate studies, so your life entirely based upon that specialization. So choose your specialization very wisely and start preparing yourself. Sorry, once you finalize your specialization, you have to work towards that to make yourself ready for it. So if you are having any doubt, or if you are not good, strong enough, if you feel, then you can discuss with your peer professors, principals, and other lecturers. You can get your doubts cleared, or you can approach any other uh, foreign based researchers or experienced researchers like us. You can reach us out and you can, hey, I have this doubt, can you please clarify me, something like that. So we are ready to help you out. So prepare yourself. Next, do research on the field of study. So once you fix your specialization, then you have to finalize, or you have to decide which field of study you are going to do. So in under pharmaceutics, again it is divided into so many categories like formulation and coating and etc. So which study you are going to focus, which research, which study you are going to focus. That's the which field of study. That's another important point. Next, prepare a brief proposal. Once you are confident to moving out, then you should start preparing a brief proposal. So whatever the research you have done here, based upon those points, you can start preparing a brief proposal to apply for a graduate study. Once you've done that, you can choose the best universities first. So start from the best, better, and good, and go on like that. So you have to choose your university very wise. That's another important factor. Although the best universities are very highly competitive, but you should not underestimate yourself. You have to give a try. Although you may get failures, that's okay. You will get experience. Why you got failed, you will come to know at the end of the day. So choose the best universities. Next, once you decided, once you pick the university, So once you decided university, you can check their website. You can find out which professor is suitable for you or which department is suitable for you, and you can start sending your applications to them. Finally, most importantly, you do not rely on anyone. You have to try your own. You can get guidance from everyone, but you should be your own decision maker. So you can approach someone to get help, but you should not approach someone with the kind of offer. If someone says, "Hey, I have a PhD position, you can come here," something like that, please do not rely on that. Sometimes they expect money from you, some money from you, or there may be some problem, or he may get some benefit from the other side. So please don't do that. Try to be yourself. Try to be responsible for your future. So overview of researchers' life, how the foreign researchers, how the foreign research life is. As I mentioned before, it's highly competitive. Yes, it's not as simple as like our Indian system. Although Indian system, unfortunately, pharmacies, we don't have much job opportunities apart from industries after B pharmacy or even M pharmacy. But foreign, it's different. Here, everything is research based, and again, it's highly competitive. You need to work smart than hard work. Yes, in India, Indian based system, we are all well theory based education, but we are not enough practical based. Luckily, pharmacy studies in India, we have better practical experiences by doing titrations, starting from the starting titrations to the in the final year formulations. You are experiencing everyday practical classes, so you're experiencing many practical experiences also. So you have to collect your hard work and make them as a smart work. So that's what you have to do in a foreign country. And yes, you will have only limited time. If you are a master student, you will have 1.5 to 2 years, and if you are a PhD, you will have 3.5 to 5 years as a max. So you are given only limited time. So you have to prove yourself. You are the best. You can bring best from your research. Definitely, you must be a multitasking person. In foreign countries, it won't work like you can do one work today and you can think about the next one tomorrow. No, you have to plan your things well ahead. You have to have a daily plan, weekly plan, monthly plan, and yearly plans. So it works in that way. So if you plan that way, you can start your multitasking steps at the same time. Especially in foreign countries, not like India. If you are moving to other city, means your college is in Srivilliputtur. If you are moving out from Chennai, you can have hostels or dormitories. You can have hotels outside. But in the foreign countries, no, it's different. There will be expensive. So for food, for making your own food, washing your clothes, everything will be a big challenge. So how you are managing your time, that's also an important, that will also come under multitasking. So you have to be fearless person. Maybe you will have some fear, society fear in India, but if you come out of the country, you will become a fearless person. That's how I experienced, that's how I learned. When I was walking in the street in Korea, one some 5-year child approached me and he asked me, "Hey, why your skin color is like brown, dark brown?" So that that 5-year kid don't have any fear. Although they know we are different from them, their race, but still they want to know what we are. That's the fearless factor. So you will become a fearless person at the end of the foreign life. You will be more independent. That's I'm damn sure. You will become a more independent person. You won't rely on anyone. So independency will make you in a good place. At the same time, it may trash you in a bad shape. So it depends upon you. The independency, how you are utilizing your freedom to become a best person is up to you. Finally, it's up to you to enjoy or stress. Like the above six factors, if you realize it, if you can able to manage those six factors, that will become a enjoyable foreign life, or it will turn opposite way. It will become more stress and it will be difficult for you to survive, and you will. I saw many people, they just quit their master program or PhD program. They went back to India saying that, hey, the food is not good, the professor was scolding me, there are a lot of blah blah blah, something like that. So it's up to you. If you want to survive, if you want to have a better future, you must enjoy the stress and live in a foreign life. At the end of the day, you will find out who you are. So, unfortunately, I sorry to say, in India we are kind of our focus is kind of diverted by multiple sources like newspapers, cinema, cricket, politics, etc. But in the case of foreign life, you no need to worry about those factors. Those factors, government will take care of it, including your health, your scholarships and all. Your only job is just study, do research, focus on yourself. So while you're doing that, you will come to know, realize, you started realizing, oh, what I am doing, who I am, how can I improve. These things you will automatically improve. That's the beauty of a foreign life. So when during research, or while you have started your graduate studies and you started doing your research, you have to have these following steps. First, meet the hypothesis. So find out the problem. So as I mentioned before, there are cosmetic industries, they're using a lot of chemicals. So that's the problem. That's the example for a problem. So you have to find out the problem and you have to create your own hypothesis, how to solve that problem. How could What are the possible ways you can improve or you can solve the problem. Next one, you should learn new techniques. In During research, you will have more and more and more. You will sit and read, so you will learn more techniques, and eventually you will solve the challenge or solve the problem. As I mentioned in the first point, should be strong in math. The world is so competitive, as I mentioned. The other countries are highly competitive. Unfortunately, our Indian students, their math level is so low, becoming so low these days. As I saw back in Korea, I was mentoring few students. I saw their maths were so worst. Unfortunately, they are not from pharmacy, but who are all from pharmacy background, they were good at math. Like our dilution calculations, our percentage calculations were really helpful. That's why I am still surviving. Although I am from pharmacy background, but last 10 years I'm not doing much research on pharmacy background. So, but still I am surviving because whatever I learned in pharmaceutical sciences back in India, that's helping me till date. Next point, you need to learn data processing using different softwares. As you all know, you may use Microsoft Word or Microsoft Excel to develop your thesis, but that's not enough. There are number of softwares coming day by day, so you need to learn, you need to improve your skills. And interpretation of data. So, as I mentioned before, once if it is an UV data, you need to know how to interpret the data and results. Luckily, our pharmacy system in India is very well in that. If you want to know about FDA or PX, our staffs are well enough to teach you well. So these are the skills you need to further improve in your graduate studies in foreign life. And approach for help. Sometimes it's okay or it's necessary to get a help from someone else, and you can give the help back. That way it works in the other countries. Next, other three parts are repeat the thrice minimum. So repeatability is most important factor, as you know, as you studied in pharmaceutical analysis in HPLC experiments. What's the repeatability? What the reproducibility and all? So you should repeat three times minimum to get the best results or the concordant results, as like you do the titrations. Then you need to document well whatever the thing you've done in a day. You have to note it down in your notebook, or you have to write it as a soft copy. So if after 5 or 6 years you get some doubt in that experiment, or someone is asking a doubt, you need to pull out your data and you need to ready to explain to them. So in that case, you have to name your folder clearly accordingly. That's the documentation. Yeah, and again, manage files and folders. So you have to good at computer to do all these tasks. And collaboration. Collaboration is a vital part in the case of foreign research life. Wherever you go, which country you go, in collaboration, you cannot avoid the word collaboration. Means that you can join with another teams. Like if you're doing pharmaceutics research, you want to do some pharmacological analysis, you can discuss with pharmacology department in the other group, and you can have a collaboration. They can then later they can provide some material, and you can do some pharmaceutical analysis for their samples. In that way, the collaboration is inevitable or unavoidable in the case of research and industrial applications. Countries like Canada, they are giving more opportunity, even in Korea, giving more opportunities to the new commerce or new research development. So the industries are more supporting, industries are funding more to the researchers. So your final goal should be an industrial application. Last but not least, entrepreneurship. So just doing research, getting your thesis published, getting a convocation, getting a degree, going for a job is one way. But these days the system is different. You need to be your own boss. So in that in that where entrepreneurs play a big role. So you learned your knowledges in your B pharmacy or M pharmacy. So use those knowledges how to make that knowledge into a product. So you need to learn those skills to be an independent, successful person. So moving forward, we are going to see how to enter into Korea or what are the necessary things needed to enter into Korea. So it's purely based on your approach. So you can approach either the advertisement placed in the websites like Korean government scholarship programs. There are two or three of them, or you can directly approach to the supervisors, approaching them with your application proposal materials, asking a master or PhD program or integrated master plus PhD program. In 5 years, you will get your PhD degree with scholarship. That's the most important part. So you have to choose your scholarship, your professor wisely. So these days, less than $1,000 is not a good scholarship per month in any country. So you have to choose accordingly. So you have to sit and read, do background analysis a lot. So to get into Korea, you need to have IELTS score or English score. IELTS or GRE scores must be higher. Although uh, Korea has their own mother tongue, but still they are expecting foreign students should have the English score. You should get your apostolate degree from HRD Department of Indian Government, and you should have a recommendation letter minimum three from your undergraduate thesis advisors or your staff members to get a better position. That's where you can enter as a graduate student into Korea. So immigrating into Canada is entirely a different story. You can move to Canada as a student, worker, or permanent resident. If you want to move as a student, you have to ready to pay a tuition fee. The tuition fee is so high it cannot be covered as a foreigner here. But in Korea, the tuition fee is completely waived off if you have English score and your prerequisite is required by the universities or the government of Korea. But in the case of Canada, no. If you are a student, you will be considered or called as international student, so you cannot get a scholarship that much. If you can get a scholarship that again that will be highly competitive, and scholarship will be preferred to the Canadian citizens or permanent resident of Canada. You can come as a worker to Canada, but again, our Indian pharmacy degree is not recognized by Canadian government, so you have to give a Canadian government pharmacy exam. So that's another story. That takes at least 2 years. You have to register. You have to appear for 2 3 exams. That again costs almost two lakh Indian rupees, and that takes time. It takes at least 2 years to get a pharmacist a license here. But your salary will be good, but you have to be ready. You must have some income in those 2 years. That's again difficult job. And last one is a permanent resident. So permanent residency, you can apply and you can get it, but you must have experience, work experience. So as you here are, as you aware, as you came across, there are a lot of advertising agencies in India. They say, hey, we can get you a PR, we can get you a job in Canada, something. Don't rely on them, believe on them. First, go to the Canadian government website called cic.gc.ca. You can get all the information, sit and read properly. You can come to know how to enter into Canada as a permanent resident. Why permanent residency is important? Once you became as a permanent resident, you will have a lot of benefits, like student fees will be waived off to $10,000 to $1,000, something like that. So there are so much advantages. You can do any job, you can do any part-time job as a international student. That's difficult to do a part-time job. So what are the requirements for permanent residency? You have to have English score, you need to have your degree evaluated, and you need to apply to the government and you will receive an invitation and you can apply for PR with lot of document, then you will get a PR. So getting a Canadian PR is not that easy. It will take time. It will take minimum 1 to 1.5 years. So you have to plan accordingly. So the take-home message for permanent residence application is not only for graduate student, but it can be, you can think it of later of your age, like after 3 years of your undergraduate or postgraduate degree, you can still want to come to Canada. You can think in that time. So considering all these factors, how the life is in Korea. Apart from the research life, I want to just explain how the regular or normal life in Korea is. So Korea is the number one in Bloomberg ratings, the number one in innovations. There are new products produced every day. Korea is the number one in OLED manufacturer, and Koreans just use the word fighting, means they don't give up. If they fail, they just say fighting and they will just move forward. They don't sit, they don't give up, they keep on try until they achieve. That's the one good thing we should learn from Koreans. And Korea has a beautiful four different seasons, starting from top. So spring season, you will get the trees get flowers first, then leaves. It will be beautiful. Then there is a summer season. It will be more humid and hot, like the temperatures reach up to 36 degrees, as like India. And it has fall season. The leaves of the plant change entirely into different colors. And the winter season, Korean winter season, the temperatures go around -20. That's the maximum. Yeah, this is the beautiful seasons Korea have. Korean food is most important in their life. How we Indians are giving more importance to the food, Koreans do as well. That's why India, especially Tamil culture and Korean culture, have a lot of similarities. Even more than 400 words with same meaning, same pronunciation exists between Korean language and Tamil language. So apart from that, Koreans used to eat more meat, usually pork meat, and they used to have a lot of vegetables. They just used to have only 1 cup of rice in a day. We used to have a lot of rice, but they used to have more meat. That's their typical food. Korea is safer, I say much safer than any other country in the world, as like Japan. So you can go anywhere, there will be everywhere, the CCTV cameras everywhere, and people follow the rules, and it's more safer. And Koreans try to live independent in their life, so they don't depend after 18 years, they don't depend on their parents, and up to their late 80s they try to live alone. They try to do their everyday task by their own, so they don't depend upon anyone. Obviously that's me. So Korea is another best country to give more importance to the physical activities. So our professors back in Korea, they encourage us to do more physical activities to avoid the stress levels. So Korea is the one place in this world to where the more marathon races happening. So they conduct races from 5 kilometer, 10 kilometer, 21.3 half marathon, full marathon 42 kilometers, and ultra marathon like 100, 200, 300 kilometers. So Korea is throughout the Korea there is a special track for everywhere for running, and some places there is a cycling pathway for whole highway. So that's why Koreans are so energetic. Although they used to eat a lot of meat, but they do give more importance to their physical maintenance. So physical maintenance is important to maintain our mental health. So that's why Korea is doing best in it. Sorry, I'm taking more than 1 hour, so I'm going to finish soon. So research life in Korea is timeline based. It needs to be processed quickly. You have to have accessibility, high-end techniques, and value to knowledge. These are the advantages being in Korea. And disadvantages are you will be given limited time because Korea is a kind of closed society. You will have limited exposure. It's purely publication based. If you publish your paper, you can survive it or not. You have to leave the country. Some places there is a low stipend. That's why I said you have to choose your scholarship wisely, and always there will be pressure. Life in Canada, Canadian life is so diverse. You can have variety of people in one street, I say. That's the beauty of Canada. That's why Canada is surviving. The Canadian winter is so cold. Up from starting from October, the winter lasts up to May sometimes. So and the temperatures go minus negative. So you have to be careful about that. Canada is a beautiful place. It has a lot of land, so it's everywhere. Most of the places it's quiet and beautiful, and yeah, you can explore a lot in Canadian life to adopt Canadian culture. Research life in Canada, you have more independency, you have enough time, you will get more freedom of speech even. You can have high impact after completing your degree, slow and steady life, and you will get a better job in the future. Everything has positive and negatives, right? So negative part of the Canadian life is slow in progress in many places slower than India. I say India is doing better in these last 5 years than Canada in many sectors, and there are sometimes the accessibility of the equipments or other facilities will be limited, and sometimes the guidance will be limited. People try to be independent, so they don't try to get annoyed by someone else in the name of guidance, and it's time consuming. Yes, the PhDs last, you cannot get degree less than 4.5 years to 5 year in Canada and even USA. Because of that, your graduation will be delayed. And the better place is it depends upon you. No place is worst and no place is too bad in this continent. So you have to think, you have to decide, you have to analyze yourself, and you have to choose wisely. So every place is better place. It depends upon your mindset. So you have to choose it wisely. So again, me, Mohankandhasamy, I'm a senior postdoctoral researcher in Department of Chemical Engineering, University of Waterloo, and you can find me in this address, email address, and this phone number. You can find me in the Google Scholar, LinkedIn, Arcid, and Publance. And I was worked as an assistant professor in Korea last 3 years. Not last 3 years. I was working from 2015 to 2018. So that's it from me, and I would like to thank the AKCP one more time, especially Principal Sir and Bro Sir for making all the arrangements, and staff members and my dear students and other faculty members, and most importantly, the management of AKCP to providing me an opportunity to give a webinar here. Thank you very much.
Thank you, sir. Thank you, Kandasamy sir. Thank you, sir.
Thank you, sir.
Really, we had a wonderful information, sir. Presentation, sir. Any question? Any question?
Any questions? So thank you, sir, for sharing your experience. So most of them they won't share their research experience, how they got, what is their role. Those things they won't share. So thank you for sharing with our students. So I had a question in your research experience.
Yes,
How much the stipend will be useful, sir,
for post
for
for
surviving,
post
sir, for surviving
for
our day-to-day life, apart from the
for
research activities?
no, for master student or for PhD for postdoc there are different varieties. So to whom you are asking?
For
To post student.
PhD research.
So for a PhD student, as I mentioned, $1,000, $1,000 is the decent salary, decent scholarship. But these days, so that scholarship you can survive, you can save some 20 to 30,000 Indian rupees apart from your expenses. But these days, the scholarships are more than $1,500. So 1,500 is the kind of average. Hello.
Yes, sir.
Yeah. 1,000 to 1,500 would be enough for a survival.
Okay, thank you, sir.
Seems like students don't have any questions. I was expecting a lot of questions from student side.
Any questions?
You can ask anything. If you want to ask in Tamil also, you can go ahead, please.
Students, don't hesitate to ask. Please ask. So there is a question in the chat box.
Yeah, and
The
just
scope of PharmD in Canada by Dr. Soumya.
the same. So PharmD, BPharm, MPharm, you have to get equivalency to Canadian degree. So you have to check your Indian degree in whether that is suitable for Canadian Pharmacy Board or not. So you have to check the Canadian Pharmacy website. I don't think PharmD is uh, acceptable here. PharmD graduation from India is not acceptable here. So again, you have to start from like pre-pharm level. You have to apply for equivalency. You have to give pharmacy exam in Canada. Then you will get the pharmacy license.
Sir, whether I had a question.
Yes, go ahead.
Is nanoparticles have a really commercial application in pharmaceutical field, sir?
There are the liposomes, neosomes, they are all approved by FDA and they are being, for example, Doxil, that's a liposome based anti cancer drug delivery system. So they are commercially available, yes.
Normally they are available in the high-end therapies, sir, but they are not that much commercialized, sir.
Yes, again, they are not that much commercialized because still many of the nanoparticle research is not being explored, especially the toxicity of nanoparticles. It's not explored very well. That's why many countries still hesitate to approve the nanoparticle-based system. But there are alternative ways they have started utilizing, like as I mentioned, the photothermal therapy and those other alternative methods, they have started using nanoparticles.
So, what are the stability issues you have faced while preparing nanoparticles?
Uh, in the lab, we will have stability issues like if your surface modification is improper, they start aggregating each other over the time period, like they become a big particle. That's the first stability issue. In the case of in vivo applications, if you're injecting into a mice, and again, in our body, we have a lot of biomolecules starting from enzymes, etc. So they get aggregated, they get degraded as well in the body. So those are the big considerations we need to have before formulating or before modifying the surface of the nanoparticles.
So normally when we are doing nanoparticle in our lab, we are facing problem in reproducibility. So every time we are getting different results. How do you overcome those things?
So that's a kind of a typical question. Like you have to, we can reproduce, but you have to stick with the protocol. Even a small 0.01 milligram of your chemical weighing difference also will impact the nanoparticle because nanoparticles are so sensitive. So you have to maintain a standard protocol. That's why there are clean rooms available in many other labs, which are all used to fabricate electronic devices. So in that clean rooms, everything is so accurate. There won't be any dust. So those are all major factors.
Thank you, sir. There is a question from Dr. Tamil Jyoti. How to know about a postdoctoral notification and what are the quality you want to apply for postdoctoral?
So that's the good question. So if you want to apply for a postdoc, the best time to apply is immediately completing your PhD, or when you are going to complete your PhD at least 6 months before. So here in the foreign life, we are planning well ahead. So we are planning at least 6 months before. So if I am graduating, this is August, right? So if I am graduating in the September, I should have some plan now. So I should start applying now. So, and after if suppose you cannot apply that time, it's okay. After your thesis, after your defense, you will have enough time to apply for a postdoc. So, after completion of your degree, you can start immediately apply it, but you should not apply more than 3 years. So, if there is a difference between after degree completion and you started applying after 3 years, means you will not get a postdoctoral fellowship. So after completing a PhD, you should start apply immediately. If you are applying after 3 years or after 4 year or 5 year, there are very less chances to get a PhD PDF fellowship. So all the labs, they need young blood. So after most of the universities, you can check in their website, most of the universities, they used to say if in the postdoctoral fellowship application, you must be in 3 years after completion of your PhD. That's the kind of standard line they used to have.
Thank you, sir. Sir, you have been in three different regions. You have been in India, you have been in Korea, and now we are in Canada. So, how you are seeing the role of pharmacists in all three countries? Even this COVID pandemic, also the pharmacists are the unsung heroes. Their duty is not recognized. Even they are also working as like other healthcare line workers, but they are not considered as heroes. So, how you are seeing the difference between these three countries in the role of pharmacists?
Okay. So in the case of Korea, I don't know much about pharmacy or any professional degrees because Korean professional degrees are only taught in Korean. So we are not allowed. Foreigners cannot learn their pharmacy degrees and to become a pharmacist? No. As like Tamil Nadu, Koreans are giving Korean language is a kind of soul for them. So that's the other story. Yeah. So I don't know much about Korea, but in the case of Canada, yes, they are giving more importance to paramedical workers, like equal importance. But again, the doctors are given first preference here also. So in India, the importance was not given to the pharmacists or other healthcare workers. That's so unfortunate. But uh, but we don't need to care. They're just doing their duty. They're just this is a service motto based job, so they're just doing their duties. That's it. This is what we learn, right? We no need to expect anything. This is our service. This is our duty to do the humankind, our fellow humans. In here also, they used to give some kind of recognition to the healthcare workers. Last time also one big burger company, they came forward to give free burger as a big uh, big lunch for every healthcare workers in the hospitals in our town, in our city. So in those way they are encouraging the healthcare workers. So in Indian government, Indian people also just uh, we have to encourage our own pharmacists by doing some ceremonies or doing some functions or giving some gift or giving just sending a card. In those ways we should be encouraged in India.
Thank you, sir. So even though you have quoted the needs of a student to pursue career in foreign countries, sir. So can you again repeat what are the important criteria to be pursue their higher studies in foreign countries?
So as I mentioned, first they need to fix their specialization, and they need to start looking. They need to do research, and they need to select professors. They need to universities first, then professors, then they start applying. Those are the initial steps. Additionally, they need to think about if you are applying for a specific professor, you need to check the professor's background, how much paper publications does he have, what are the fundings, what he have, and how good he is, what is the citation, how much citations does he have. These are the other important parameters you should consider. Just going to the some ABC lab or ABC professor, joining, completing a PhD, that won't help because you are from pharmacy background, you are already having a professional degree. If you're going to some XYZ professor or XYZ lab, your pharmacy degree will be wasted. So don't do that. So these are the other preliminary factors you need to consider. And you can, if you are living in a perfect way, you can save better money and you can send back to home, or you can save it for your future studies or future living as well. Those are the things you can consider.
Thank you, sir.
Uh, Thank you, sir.